Okay, what I want to do today is continue the thought process of talking about sort of the biochemistry of the cell. We did cell respiration. Now I want to talk about photosynthesis. I want to approach it from a couple of different ways, and that'll be today. I'm not sure yet what I'm going to do tomorrow. I have a couple of things in mind. I just have to wait and see where we get and so forth. I didn't get as far this morning, but that's okay. We had a shorter period of time, so I don't know. Okay, where I want to start with photosynthesis is basically sort of with a historical approach for something different, and that is an experiment carried out by Van Helmont. Van Helmont was, obviously the clicker stopped working, it was interested in this concept of where does the biomass of a plant come from. I mean, he observed that he could start with a seedling, and then you can go from a seedling that you could hold in the palm of your hand to that's a person to a fairly large tree, and where is all that biomass coming from was basically his question. So he conducted an experiment. He was, as you can see, this was back in the early 1600s, so it's fairly long time ago. He conducted an experiment. This is the experiment written out. I have a diagram in a second, but basically what he did was take a seedling, plant that seedling, water that seedling, watch and take care of it for 5 years, and then harvest. Well, first he weighed both the pot of the soil and the plant itself. Then he took care of these plants for 5 years, and then he weighed the plant and soil again. In other words, he planted, let the tree grow, then separated out the plant again to determine the weight of the plant. Now, the only thing he added was water. So he took his 5-pound willow sapling. It grew to almost 170 pounds. He started with 200 pounds of dirt and ended with just about 4 ounces less than that, but practically the same amount of dirt, soil, I guess I should call it. And obviously the tree grew quite a bit. And his conclusion was that I only added water. It took nothing from the soil, so the weight must be from water. Okay, that was Van Helmont's conclusion. That is that water provided that. What I'm going to ask you, and this morning I did it, we had a moment of silence as no one would answer, so I'll try to duplicate that moment of silence again now, is, think you can discuss it. Sorry, this bubble's a little messed up, but this round table of students is thinking about, well, maybe some of the tree came from the minerals in the soil, but, I think plants make food from sunlight. Maybe Van Helmont didn't know about gases in the air, or I agree with Van Helmont that water is probably what made this difference in the biomass of the plant. What do you think? I didn't ask whether you cared. I asked what you think.
A little bit of everything.
A little bit of everything. That's a good diplomatic way to look at it. That is, it's a little this, a little that. Okay. Yeah?
It's from the air mostly.
From the air mostly. Okay, so we have at least a diplomatic and a sort of solitary answer here that it's mostly from the air. Anybody else?
Soil, sunlight, air.
Well, I mean, I don't know how much sunlight weighs, but that could increase to increase the weight of a plant that much. But okay, the soil, it took some of the soil. We lost about 4 ounces over a period of 5 years. Well, if I gave you just a little bit more data, this data, not his data, but more a modern approach to looking at a plant, maybe this would help us too. That is, the idea here is what this was a series of experiments where plants, so various plants were taken and subjected to different concentrations of carbon dioxide. Okay, so basically put in a chamber and 40 parts per million carbon dioxide, 80, 1200, 1600, and then allow the plant to grow for a certain, in all cases, the same amount of time, whatever it was. I don't recall at this point whether it was 4 weeks or 2 months or whatever. But allow the plant to grow, harvest the plant, and determine the dry weight. So this is sort of growth of the plant or dry weight of the plant. And what you see here is what? Why am I interpreting this? Okay, roots show a plateau. The effect of carbon dioxide on roots, there's no real effect that we can see. I mean, the plants grow, but certainly plateau, the carbon dioxide doesn't matter. Does that make sense in what we know what plants do, or plant roots do? Why?
Because they get their nutrients from the soil more so than they get from.
Well, they get nutrients from the soil, but that really doesn't explain that here. That doesn't apply here. That is a true fact, okay? But that doesn't explain this flat line here. What do roots do?
Go down.
Well, what cellular process do they use?
Absorb water and nutrients.
I spent 2 days talking about it. Yeah, cellular respiration, right? They respire. So adding carbon dioxide to a system that respires wouldn't really make any difference at all, right? And so that makes perfect sense that the roots, that makes no difference here. This little bit here, I got asked, why would that start? Well, I'm not entirely sure because I don't know all the details of the experiment, but it could well be that there was a little bit of microbial respiration, which then is inhibited as CO2 goes up, but that would have released maybe a little bit of nutrients and spurt of growth, then that would level off. But certainly over a wide range, these roots are respiring, so it doesn't matter that there's CO2 there. That makes sense to me. This is CO2, not oxygen. Okay, if we look at total plant, what do we see as far as the trend of total plant? Does CO2 concentration matter there? Yeah, it seems to matter quite a bit, right? The more CO2, the total increase in plants, the greater the concentration of carbon dioxide, the greater total plant biomass. That works. They separated out shoots. I wish they had done leaves as well, but these were herbaceous. These weren't trees, so we're not worried a lot about respiratory mass of bark and so forth that don't photosynthesize. So these were herbaceous plants. They, I believe, were mung beans, I think, but I'm not entirely sure. Because that part didn't matter to me, so I didn't keep that. But the idea here is that we do see an increase in the production of biomass, both of the total plant and of the shoots, with increased carbon dioxide. Okay, sure.
Doesn't every cell of the plant respire, even the photosynthetic parts?
Okay.
Well, they do, right?
Yeah.
Okay.
Oh, okay, I thought you were going to take it another step. Yeah, okay, I was just going to accept that and waiting for the next, the zinger on that, but okay.
You just suggested that the roots, I would have thought that the root level would also go up just to support the additional shoot mass. I would think that there'd be a slope on that root mass, all, along with the shoot mass, and the total would, obviously be a steeper slope. Just because the whole thing is respirating, and I would think that more shoots demand more roots.
Well, to a certain extent, I guess that's true in this system because, again, it was an herbaceous plant. I'm pretty sure it was mung bean, but okay, like a broad bean, like a lima bean, sort of similar to that. They I won't say have determinate growth, but they're not like a tree that continually adds all this excess respiratory biomass, which the roots also have to support as well. So it probably is an artifact of the species that was chosen, and so I do apologize for not having that species listed on here, right? It's not anywhere, right? I'm always usually, I'll say that and then somebody will point out, yeah, down in the corner it says it's mung bean. But so I see what you're saying, but so that wouldn't change the overall trend, but it might go up a little bit, yeah, because of the overall growth of the roots. So that's quite possible. I think it's more of an artifact of the species that they chose, and that wouldn't really continually add that much of root biomass in the time frame of the experiment. Okay, that I'm pretty sure would be the case, but the overall trend is what I wanted you to see. That is that these are photosynthetic biomass, again, because they are herbaceous, both the stems and the leaves would photosynthesize. That's important to understand. So we do have increased availability of CO2 permitting increased growth. That is CO2 enrichment increasing photosynthesis, increasing the production of biomass. And that's a fairly substantiated relationship that we see. Now, obviously, nothing, you're going to have a law of diminishing returns, and this will ultimately level off, and that's the case. Someone asked me this morning, is this something that would be done commercially? And the answer is yes. That is, not your local florists, but large greenhouse production facilities do pump carbon dioxide into the greenhouses to enhance production. I know that they do that in carnation production, because I toured a greenhouse facility outside Bloomsburg where they did that. They're the largest East Coast producer of fresh carnations, and the greenhouses are so large they can't ventilate them, to get fresh carbon dioxide in there, so they run tubes and not saturate the air, but add carbon dioxide. They know exactly what amounts to add. They look at here and say, well, this is about what we want to add, because it costs money to add it, so they only go as far as they need to. But yeah, it's done. Okay, so we get this enhancement. So why I think this is relevant to, Van Helmont didn't have this, but if he had this, perhaps he would change his tune a little bit. He thought only the water was important, but it looks to me like gases. And, unfortunately for him, or I shouldn't say unfortunately for him, it was partly his personality, so I understand from reading about him, he really didn't care what other people thought. That is, I don't mean that he was an independent thinker in the sense that he wasn't ruled by fashion or anything. What I meant was he really didn't care what other scientists were doing, even though some of them were starting to work on atmospheric gases and the importance of it. He didn't really care to read that. He was doing his own work, which was far more important than these other people's work, and so he would have benefited from a little collaboration, but he was too arrogant for that, apparently. And because apparently when he would publish a paper, and if somebody would disagree with him, he would challenge them to a duel. So I guess after you do that a couple of times, people don't really want to collaborate very much. So he, in fact, died in if I understand correctly, died in a duel. Okay, so he needed to be a little better at shooting. The other thing is that it does turn out, this is not perhaps the best photograph for proving this, but I just want to suggest to you that here's a little experiment, and in these cups was just plain water, and in these cups were water with a little bit of nutrients added, and it's a little hard to tell here, but these plants are growing better, so we know that nutrients are important. In fact, Van Helmont had to know that because he was a gentleman farmer among other things. And he knew as well as anybody else that that plants benefited from fertilizer, from manure, from the farm and so forth being put on the fields. So he should have known that nutrients might be important here as well. So we've got three things here that I think we need to be concerned when we start talking about where wood comes from, and that is that it's certainly carbon dioxide from the atmosphere, and we'll see obviously that's the bulk of it. In fact, that water certainly plays a role in this, and without it the growth would be impossible. Not only because plants need water, but photosynthesis consumes water, as we'll see. And you really do need some nutrients, although they may not necessarily add to the bulk, or I should say to the weight of the plant, but they certainly facilitate the growth of the plant. So the real answer, I'm not going to ask you to do this because it was fun this morning. Now it's just annoying. The answer here is that most of the increased weight came from the air, and then probably some of the water. And yeah, maybe a couple of ounces of essential nutrients over the 5 years would be the best answer there. So it's all of the above, okay? So you were sort of right, but it's mostly carbon dioxide, so he was right as well. So see, everybody wins, okay, in this, which is the way it should be. The only other thing that I wanted to say before I left Van Helmont is that he is a good example because most of his work was disputed later and in fact proven to be wrong, not just disputed. And so it's nice to know that he's still remembered, even though most of his work was wrong. And so it might be a good example to draw some conclusions from. And one of it is that you don't want to ignore the data. That is, he knew that, for instance, that the soil had changed, but he dismissed it, because he was focusing on the water. He wanted to prove that. I mean, he had said that from the outcome of his experiment that he believed water was the only ingredient needed, so that sort of blinded him. And you don't want to do that if you're doing an experiment. And the other thing is that you really do need to collaborate. That is, you need to be aware of existing or pre-existing or coexisting knowledge, because as I said, he knew about this idea of nutrients being important, but it didn't matter to him. And I guess the other thing that's interesting is that in science and history, it turns out that being first is what really matters. Because even though he was wrong, he was the first one, so he gets remembered. And I bet you can't tell me any of the names of the people who disputed him. And actually proved, came up with the correct answers. And I'm not going to bother to share them. The only one I'll share is Joseph Priestley, and you've probably heard of him. He's sometimes called the father of modern chemistry. He's supposed to be the discoverer of oxygen. I say just supposed to be. He is credited with being the discoverer of oxygen, of the carbonation process. And he also worked with plants, but he was about 75 to 100 years later than Van Helmont. And he worked with plants as well, and he was able to strongly implicate that plants needed light for photosynthesis, in addition to carbon dioxide and water. So, and I mention him because he's a local boy. He lived in Northumberland, up along the Susquehanna, and I visited. You could go visit his house. It's interesting. I wouldn't. It's not a destination. That is, I wouldn't go there to Northumberland just to visit Joseph Priestley's house. But if you're in the area, it's worth stopping by. They give guided tours. We had Helen was her name. I think she dated him. I'm not sure because she's that old. But she was a really good docent and took us through. And they're doing excavation there and restoration of the house. It's really pretty interesting. I also learned, and then I'll leave Joseph Priestley. The reason Joseph Priestley is dear to my heart is that in 4th grade, that's the first time I remember, because we're back to old, really long time ago. First time I ever had to stand in front of a class and do an oral report, and I did it on Joseph Priestley, about him discovering oxygen and all. And so That's why he clicks with me. I don't know why I picked Joseph Priest. I was a nerd, but still am. But anyway, the other thing I didn't know about him is that he was a contemporary and a good friend of Ben Franklin and Thomas Jefferson and some of the other authors of the different constitutions and so forth at that time, and really had a lot of influence on them. His ideas apparently were very radical, so they really didn't want to admit they knew him. That's why he lived up in Northumberland, and they go visit him and talk about it and then sneak back down to Philadelphia. But, it's really pretty interesting. I didn't know that. So he's sort of a ghostwriter of the Constitution, let's put it that way, and of the Declaration of Independence as well. Okay, so that's our history lesson for that. Okay, so I'll come back to some of that in a moment or two, but I want to then talk more directly about photosynthesis for a little while here, actually for quite a while. If you recall, when I did the same thing, I keep hoping that'll work. The first thing that I can mention, although I don't like summary equations for respiration or photosynthesis, at least I'll start with that because we know that water and carbon dioxide are essential, as is the sunlight. Here we don't mention anything about the nutrients. And then the product is oxygen and carbohydrate. It says here glucose. I know that a lot of textbooks say glucose, but that's wrong. It's not true. Photosynthesis does not produce glucose. That is not the end product of photosynthesis. Never has been, never will be. So I don't know why it's promulgated in textbooks all over the land that this reaction and that's glucose and that's what's formed, because it's not true. Okay, it's check me, it's not true. But it is true that water and carbon dioxide are used in the presence of light. Carbohydrate and oxygen are produced in photosynthesis. That much I'll buy. This is respiration, and basically, I really will skip to the next slide. I used this slide when I was introducing respiration, and again, remember, I like the idea that we're tying these two processes, photosynthesis and respiration, together with regard to carbon movement in the carbon cycle, and the idea that oxygen is being produced and consumed. So I find both in the oxygen cycle and the carbon cycle that to be good. But this idea of linking these any more than that and trying to make them look like they're sort of reflections in the mirror of one another, that is, one set of reactions versus another, is just flat out wrong, and they don't even take place in the same part of the cell. They don't really have they have a few things in common, but not that much as far as I'm concerned. Okay, when we look at photosynthesis, we realize that there's a couple of things. First of all, there's a special site for photosynthesis, at least in eukaryotic and terrestrial plants. In most eukaryotes, the photosynthetic cyanobacteria, a little bit of a variation on a theme. I can talk about that if you have questions, but I'll just leave those alone. I'm going to deal mostly with just our typical sort of photosynthetic plants. They use chloroplasts. Chloroplasts are found in the leaves of the cell, or in the cells of the leaves of plants for the most part, although, as I said, a lot of plants use the stems for photosynthesis as well. So wherever it's green, there are chloroplasts, there's chlorophyll, there's chloroplasts, and we can do we can have photosynthesis take place. Those chloroplasts have a unique structure internally that's certainly important, and just a couple of things we need to remember about that. That is that inside there is a liquid. That liquid is called stroma. That stroma will be important in the light-independent reactions of photosynthesis. And embedded within that stroma, that liquid, are these stacks referred to as thylakoids. Grana are stacks of them, the individual thylakoids. They become important because you see they are basically membrane sacs. There's membrane-bound sacs stacked one on top of the other. So this is a bilipid layer, bilipid membrane. What's going to happen here is we're going to move hydrogen ions in here and then run those hydrogen ions back out. We're going to use that as the site of chemoosmosis. So it's analogous to what's taking place in the mitochondria. Only the mitochondria uses that convoluted inner membrane for that to take place in the intermembrane space. Here, there's separate packets, and the protons move back and forth across those individual membranes. Those cycloids are the site of what are called light-dependent. You may know them as light reactions, but the light-dependent reactions of photosynthesis. The light-independent reactions take place in that liquid stroma, and that's all packaged in a double membrane as well, as this shows. There's an outer and inner membrane, and then the cycloid membranes. And that's where light-independent reactions take place. So we have this sort of double take that we need to be or sort of separation that we need to be concerned about. Like I said, unfortunately, this uses light and dark reactions, or maybe not. It gives I learned it as light and dark reactions. I still hear people say light and dark reactions, but obviously they don't occur in the dark. They occur simultaneously. This one intimately involves light. This one tangentially involves light. This one's light dependent. That one's independent of light, which is why they're given those terms. They're both going to be in the leaf. This summarizes the idea that within the light reactions is where where carbon dioxide and water are consumed and oxygen is produced. And the light independent reactions are going to be where carbon dioxide is consumed, and I think I said something wrong. No, okay, carbon dioxide is consumed and sugars are produced. If I did, it'll come back to haunt me. As I said, if we looked at an individual thylakoid, that's what this gray is supposed to be, an individual thylakoid. What we see embedded in the membrane here is, again, an old friend, ATP synthase, which we've talked about. This is the protein structure that permits protons to fall through a gradient, spin an armature, and produce ATP. So it's going to allow chemoosmosis or enable chemoosmosis. Also embedded in the thylakoid membrane will be two photosystems. Well, two types of photosystems designated photosystem 1 and photosystem 2. Talk about those in a second. And joining those two systems will be, here it's just shown as a green box, but there's a complex of several cytochromes which are going to be specific pigments that join photosystem 2 to photosystem 1. Okay? Let's talk about those photosystems just briefly, if we could. Imagine the leaves of a plant as antennae, which are trying to collect solar radiation. Within those are going to be microantennae, little collecting dishes. Those little collecting dishes come in two varieties, as I've said before, photosystem 1 and photosystem 2. These little collecting dishes within the chloroplast, actually within the thylakoid membranes of the chloroplast, look nothing like this, but basically consist of a cluster of pigments. In this case, we could think of them as chlorophyll pigments. How many? Between 250 and 500, somewhere in that neighborhood. It depends on species. It depends on light exposure, so forth. The long and short being that this particular cluster of pigments are connected to one another. Adjacent and connected to one another, and each one has a centrally located, it doesn't have to be in the exact center, as it shows here it's off to the side, but has a centrally located structure which is a modified chlorophyll, a modified chlorophyll. It's called a photosystem. Well, excuse me, the whole thing is called a photosystem. That modified chlorophyll is called a reaction center of the photosystem. Like I said, the reaction center is a modified chlorophyll. How is it modified? Why? Well, it's modified to attach to the membrane and to transfer electrons to the cytochrome chain. So it's just got a little bit of a rearrangement of a couple of the side chains. as I said, there are two of these systems, or two kinds of these systems, PS1 and PS2, designated by the way they were discovered, so the numbers don't really mean anything, they're just so we could have different numbers. What's important is that they are designated, they are different and designated such because they have different reaction center peaks. That is, as I say here, photosystem 1 has a reaction center whose peak absorption is at 700 nanometers. Reaction center 2 of photosystem 2 has a peak absorbance for the phytochrome in the center, the reaction center, of 680 nanometers. Now, if you remember, they're pretty close together. P700 for photosystem 1, P680 for photosystem 2, and they're both very close in the red range, but they're in the red range. And this is absorption, so they're absorbing in that red area of the spectrum. That is, I guess, for now, I'll say it, then I'll sort of contradict myself. That's sort of the optimal wavelengths for photosynthesis at this point. That is, that sort of red is sort of the center, the bullseye that the plants want. They do, though, as you can see, with this sort of, with just the visible spectrum, let alone the entire electromagnetic spectrum, but certainly operating within the visible spectrum, that if they focus just on that, they're missing a lot of other energy. Well, they have a way of dealing with that. They're going to, let's see, I'll come back to that in a second. Oh, I guess I'm jumping ahead of a couple things here. Well, I'll jump around and come back. They have a way of dealing with that. That is, they have other pigments. Remember, just the 2-second definition of a pigment is a molecule that absorbs or reflects or transmits, depends on which, but absorbs a light of a specific wavelength, therefore it gives us a color. If you were to take leaves, I guess they're beet leaves, I don't know what they are, chew them up and put that on chromatography paper, dip this into a little bit of acetone. After a while, you'd see these pigments migrate, and you could see that, yeah, there's a lot of green pigment, but there's some other colors here as well, and these are named. Those pigments typically are called accessory pigments, but that's really not fair because it makes it sound, when I think of accessories, I think of, things that don't really matter, a belt, earrings, a ring, whatever. They're accessories. You're accessorizing. Well, that sort of diminishes the importance of these guys because they're far more important than that, because each one has a specific absorption spectrum. Now, the green one is as you can see, is the P680, P700. Those two sort of combined together. Really a narrow peak that would be usable by a plant, but by expanding using these accessory pigments, the plant is able to use a broader spectrum of the visible light waves for harvesting energy, which is what it wants to do. So it's going to be able to absorb more light waves from that particular than just the P680, P700 peaks, which would be a little problematic. I don't know. Okay, why there isn't some accessory pigments that pick up here? Does anybody know? Because I don't.
I remember just reading something like basically plants are adapted to select the red wavelength for their quantity and towards the blue end for their quality. Just in between the,
Okay, I mean, that sounds good. Anyway, I honestly don't know. I'm not sure what that. I'm not quizzing you. But I'm just saying I'm not sure what that means to say this is quality, and this is quantity.
yeah.
I don't know what that means. I mean, I know, okay, that we're if anything, this is less quantity because it's decreased energy. So I don't know. Okay, so I don't know. That's why I say I don't know why there's this dip here. It would seem to me, well, maybe God made it that way. Okay, because I don't know why there wouldn't be some pigments that would pick up there too, but there aren't. Okay? So I've been asked that many times, even way back in the Dark Ages when I taught botany. I would be asked that, and I never saw an answer to why there's that. I understand why you have absorption peaks. I understand how you can expand those peaks with accessory pigments, but why they left that middle part of the spectrum out, I don't know. Maybe they just didn't like those colors. I don't know. Anyway, so those accessory pigments, this little model makes them all look like they're the same, but these accessory pigments would be located around the reaction center. They would be able to absorb other wavelengths of light. What's happening then is that the energy, because remember light can be envisioned as packets of light, as photons, and those photons are gathered by these different clusters and then going to be focused to a single reaction center, P680 or P700, depending on which system we're dealing with, and then the energy will exit this photosystem via that reaction center and be sent into the cytochrome chain, which is where I'm going next, okay? This is the cytochrome chain. I'll come back to that. I don't really like that diagram too well. Actually, that's not the cytochrome chain, but I don't like that diagram, so I skip it. I just put this here to indicate how those photosystems are embedded in the thylakoid membrane. This is the thylakoid membrane, this membrane. You can see here's the photosystem here, here. I assume they have them designated photosystem 1, photosystem 2, green and gray, just to show there's two different ones. They're obviously not green and gray. And they're embedded in, and they have these double loop proteins to hold them in place, which isn't uncommon in cell biology. That is, if you want something to be stuck in the membrane, to have these double loop and 7 loop, and they have different names of proteins that rope in and through the membrane, sort of back and forth zigzagging, and then you can anchor things inside the membrane better. Because remember, because I keep thinking of things. Because remember, these may or may not be hydrophobic or hydrophilic, and so you need a way to actually anchor them. So you use these big winding proteins that are sort of wedged in there, and then you can attach things to them. You had a question.
If we go back into the visible spectrum, the one for the peaks. The one for the peaks and the accessories.
Not that one then. I don't know which one. This one. Okay.
So I'm trying to figure it out. What's the whole purpose of just doing a visible spectrum here, just to quantize what?
Well, because this is what the plant absorbs. It does not absorb in the ultraviolet. It does not absorb appreciably any more than we do the infrareds. So it only absorbs in the visible spectrum, just like our eyes only work in the visible spectrum. Plants only work. There is no plant, like insects can see in ultraviolet light. There is no plant that uses ultraviolet for photosynthesis, that uses ultraviolet light for photosynthesis.
So basically, we're talking about for the PS1 and PS2, those two absorption wavelengths are just characteristic to photosynthesis?
I'll let you finish. Go ahead.
So, because I'm trying to figure it out. Okay, so each plant, doesn't matter what plant you do the absorption, you're always going to find these two peaks in that visible spectrum. Yes or no?
You're always going to find P680 and P700, correct.
Those are the
Except if you go into certain, blue-green algae and so forth. They're a little different because they're not really eukaryotic and they don't have thylakoids and so forth. But if we stay in oaks to maples to sugarcane to marijuana to, I don't know what.
Labels. You can call it like labels of photosynthesis.
They're not labels of photosynthesis. They are the actual molecules that there is this. The plant is like a radio dish, like a satellite dish, collecting sunlight. Okay. It does that at the level of a leaf by putting the leaf out and letting the light shine on it. Within the chloroplasts of the leaf, within the cells of the leaf, and then within the chloroplasts of those cells of the leaf are these tiny dish-like structures. They're really not dish-shaped, but these clusters of molecules called photosystems, two different varieties. One called photosystem 1, one called photosystem 2. Photosystem 1, P700, photosystem 2, P680. The idea is that what differentiates those two is this reaction center. One has a reaction center, the molecule reacts at has an absorption peak at 680 nanometers. The other has an absorption peak at 700 nanometers. Now, that's what's used to distinguish the two. Now, functionally, I haven't gotten to how they functionally differ, but to distinguish these two. So in all the world of all the plants, there are only two varieties. It'd be like Chevy and Mazda. There's only two kinds of cars in the world. There are no other kinds of cars. PS1, PS2, 680, 700. There are no other ones in the world, at least not that has been discovered yet. Now, functionally, what they do will also distinguish them. But how they're distinguished here would be like the red Mazda, the blue Chevy. Okay? In this case, it's P700, P680. Okay? Does that help? No. Or is there, what else?
So basically what we're doing with this absorption is to quantize how many PS1 and how many PS2, basically?
Well, we're not really...
I'm just thinking in terms of...
We're more classifying them than quantizing them, but sure, we're organizing them into these two centers. Correct. Okay? Mm-hmm. Yeah. Anything else? I mean, I'd much rather answer your questions than listen to me. I don't know how you can stand it. Okay. You don't. I really don't. Okay, because I know I couldn't. Okay. I don't know, again, whether you want to do this for real or not, but here's the question that comes to my mind then. Actually, this was a student-generated question. That is, okay, you're, a very respectful student, Dr. Pratt. Okay, you're saying, Dr. Pratt, you said that the idea here is that the plants want to expand the amount of visible light that they can harvest, and they do that by having these accessory pigments, the carotenoids, the xanthophylls, and all those kinds of different things, and that funnels into these reaction centers and improves photosynthesis by allowing them to absorb other wavelengths of light that they wouldn't be otherwise able to do. So why not make them black? Because black absorbs all wavelengths of light. I thought that was a really neat question, okay? I mean, I think there's a pretty simple answer, but I thought, hey, that kid's actually really bright, although maybe he was just really dumb, and I just gave him more credit than he deserved. But I thought that was a really thoughtful question, at least at first. I mean, he might have come to his own answer if he thought about it a little bit more. Well, what do you think? I mean, I'm providing some suggestions. Maybe I shouldn't have provided those suggestions because this morning folks came up with one other that I hadn't thought of. Mm-hmm. What do you think?
Should we use our spins?
If you want to use the clicker or just shout out or go, okay, that's fine too. I don't care. I mean, I know. I was all into these clickers, but they got old fast. Okay. So what do you think? Because doesn't it make sense? I mean, because white would reflect everything, so you wouldn't want that, and black would absorb it all. So if you made a black pigment that you could, again, you'd have to go on faith that you could connect it to the photosystem and so forth, and I'll give you that could happen. Plants still wouldn't use it. Why? It's too damn hot, okay? It would cause a thermal overload. And that's a real problem for plants. Plants really have to worry about temperature. You think about it for a couple of reasons. and I'll sort of take a little detour here, because remember what's going on in a plant is transpiration. And transpiration is this movement of water from root to shoot to leaf and out of the leaf through evaporation using that cohesion-adhesion tension, which I explain a million times and I still don't understand how that can possibly work. That is that a molecule of water evaporating from the leaf has enough tension to pull the next water molecule or the water molecule next to it, which then pulls the next water molecule next to it, which pulls the next water molecule next to it, all the way down the petiole of the leaf, down the stem, down the trunk, into the roots, and through out to the root tips to draw water in through the tip of the root hairs of a root. Okay, I mean, that's exactly what happens, and that's exactly the explanation, but does that really, I mean, okay, but that's what happens. Okay, it's the plants do a little trick, some tricks to that. They have special cells, xylem vessels, and tracheids that have specialized designs. They're just specialized tube structures to prevent cavitation, that is, air bubble introduction. And if an air bubble is introduced, the tracheids and vessels are designed to bleed those bubbles out so that they reestablish that water column. But that's transpiration. Now, why that's important or germane to my ranting and ravings here is that this is this plays a vital role, first of all, in drawing nutrients up through the plant. It's the only way to get nutrients from the soil to the top leaves. And think this is working in a sequoia or giant redwoods. I mean, it's unbelievable to me. It's solar powered. There's nothing more to it than solar power. Shut the lights off, transpiration stops. Turn the lights back on, it works. So it's solar powered. So it does pull nutrients up. It also cools the leaves. Okay? So plants worry about temperature. Temperature affects photosynthesis. I have a diagram in a second. The water exits through these stomata, through these openings in the leaves. That's the good part. The cooling would work from evaporation because that's just, the water molecules that can escape will have the highest kinetic energy. They take that kinetic energy with them. What's left is an average lower kinetic energy. Average kinetic energy is temperature. So if you let the high energetic molecules go away, you cool the leaf, or you cool the skin or whatever. So evaporation is pretty easy to understand in that respect. The problem though is, of course, that you're going to cool this down, but you need that water. You absolutely need that water because living things need water for osmotic balance and so forth. These plants also need this water because they literally consume the water in photosynthesis, as we'll see in a moment. So that's vitally important. The other problem is that if they lose too much of this water during transpiration, they could dehydrate and die. And you've seen plants wilted and so forth. If not, I'll come to my house. I'll show you lots of wilted plants on the front porch and so forth. I said that temperature affects the rate of photosynthesis. This is just a sort of generic little graph that sort of represents that. Here's temperature along the x-axis, rate of photosynthesis along this axis. Initially, we see, because this is a Q10 reaction, that is, this is an enzyme-controlled reaction, you do get an increase reaction rate with increased temperature. That's pretty standard. You do get that. But you do reach a point at which, if the temperature gets too high, photosynthesis rates start to drop off dramatically. And that's what you see here. And notice it's not super, super hot. We're talking, what, 30, that's 30, so right around 30 degrees Celsius, which I should have done the math, but I didn't. 24 is about, room temperature is about 72 or 73, so 30 can't be more than 90 degrees, if it's that, somewhere. I don't know.
82 is 28.
82 is 28, so there you go. So I was closer than I thought. So yeah, so 30 is a little higher than that, so we would be pretty good. So, well, and 37 is 98.6. So you can see that's not super hot, and you start to get a real drop in photosynthesis. So plants have a real balance that goes on there. Now, as long as they've got plenty of water, it's not an issue, because they can just ramp up transpiration, keep these leaves cool, keep photosynthesis going. Moving materials, great, life is good, but as soon as there's a problem with water, and typically there will be a problem with water, even in unless you keep your feet wet, like mangoes or mangrove trees or something, you're going to run into water problems. You really are. And what follows after water problems are temperature problems. So my point here is a long story. I do that a lot, give long answers for short questions. You can't use black pigment because you'd end up with a thermal problem that would be untenable. You just wouldn't be able to do. I'm sure you could kill just about any plant if you sprayed it with a non-toxic black paint. It just wouldn't make it. I mean, even if you sprayed it such that you didn't clog the stomates or anything, I think the heat load itself would be an interesting experiment, but I'm pretty confident how it would work out. Okay, I want to focus just a little bit then on the light reactions, then we'll take a break here. The light reactions are typically I should call them light-dependent reactions. See, it's my old school coming in, are typically presented in this what's called a Z scheme. Just because it's diagrammed as a Z, that's basically the reason it's called that. And it's diagrammed as Z to try to represent the energy relationships that take place during these two photosystems. So it's all arbitrary. There's no Z in the plant itself. Okay, here are the two photosystems. They're shown twice because this is the low energy state, this is the high energy state. Low energy state, high energy state. Okay, that's why there are two of them, two of each of them. I'm going to start over here with photosystem 2. Again, the reason that's 2 is this was discovered first, then this one. So this one was numbered 1, this is numbered 2. And that's it. There's no other reason for the numbering. But some critical things happen here in photosystem 2. That is, again, light hits all those molecules, the photons are collected, they're shuttled to the phytochrome 680, that is the reaction center. At that point, energy is extracted. God knows how that happens, but energy is extracted from that electron to cause photolysis of water, as you can see indicated here, releasing oxygen and protons. Those protons are going to be pumped across the membrane momentarily, but we have photolysis taking place at this point. So the chemical breakdown of water taking place under the direction of photosystem 2. Absolutely critical, because without that we wouldn't have the hydrogen ions to pump back and forth, and we wouldn't make, chemoosmosis couldn't take place. That causes an energy elevation because we've absorbed energy, so the energy content of the phytochrome is high. It discharges that electron into the cytochrome chain. These are the individual cytochromes of the chain. There's actually a couple more. The idea being that just as in the electron transport chain of photosynthesis, the energy is extracted carefully from the electrons as they're passed from one cytochrome to another. What's done with that energy is to drive protons across the thylakoid membrane into the inner thylakoid space and accumulate hydrogen ions. Then the electrons, now in a lesser energy state, are added to photosystem 1, at which point light's shining on this. Those electrons are boosted up to a high energy state. In this case, the electrons are then added to, we saw a similar molecule to this in the mitochondria, the nicotinamide adenine dinucleotide. In this case, it's got a phosphate group on the end, so it's nicotinamide adenine diphosphate, or nicotine adenine dinucleotide phosphate, but it's been reduced here, so the electrons end up here. So these electrons go through this, energy extracted for proton pump, then boosted again and used to reduce NADP. So the light reactions produce oxygen. They produce, as we'll see, through chemoosmosis, that would be the next step in this, ATP and reduced NAD. That is a source of reducing power in NADPH. I don't care about that. Here, just this is the same thing again. There's photosystem 2, photosystem 1, photosystem 2, cytochromes. They just didn't put them all here this time. They rearranged them a little bit, but it's showing the same idea. The only reason I included this was that, okay, we've pumped these protons across inside to the inside of the thylakoid. Now we let them run back through the synthase, spin the armature, produce ATP. So the products of the light-dependent reactions of photosynthesis are oxygen as a waste product, ATP through chemoosmosis, and reduced the reduced NADP to NADPH. And those substances, the oxygen is a waste product, okay, which we really need to talk about because it's problematic as it builds up in the plant tissue. It's actually problematic for photosynthesis. Too much oxygen is bad, and we'll talk about that in an hour. And then the other thing that's produced then is the ATP. Okay, that shows the same thing again. And again, I had like 3 or 4 of those. So the idea here is the light-independent reactions will provide ATP and reducing power for the light-dependent reactions. Now, let me just set this up. The light-dependent reactions.
Again, you said the opposite of what's on the slide.
I did? Okay. The light-independent reactions produce ATP and reducing power for the light. Yeah, I did it again, didn't I? The light-dependent reactions produce ATP and reducing power for the light-independent reactions. Okay, I apologize for that. Thanks for catching me on it. Okay, that's why it's written here. Thank you. Okay, the Calvin cycle is an alternative name for the light. See, that's why I'm doing this twice, doing this for 3 hours, and doing this is just hazardous to my health. And I'm not doing it anymore. Okay, this is it. Okay, and no offense to you, you're all good people. I'll miss you, but I don't miss doing this. I apologize. If David will let me resign. I've tried to resign once before. Okay. But anyway, the point here is that an alternative name for the light-independent reactions of light-independent reactions, the dark reactions of photosynthesis is called the Calvin cycle, again, named after Calvin Benson, Benson Calvin, his last name is Calvin, who discovered it much, sort of like with Krebs, George Krebs, and the Krebs cycle. So that's what's going on there. So as this little diagram suggests, we've got ATP and NADPH coming out of the light reactions, that is the light-dependent reactions, and going to the light-independent reactions of the Calvin cycle. And I want to talk about that in a moment. We're going to take a break, and then we're going to do that. And coming back from that then, because it sort of cycles back and forth, then because we use up ATP in the Calvin cycle, the ADP comes back to be rephosphorylated, and the NADP, the NADPH loses its electrons, so it becomes oxidized back to NADP, and it comes back to the thylakoid membranes to pick up more electrons. So they shuttle back and forth. not unlike what took place in the mitochondria. So there are some overriding similarities between what happens in a mitochondria and what happens in a chloroplast, but they are separate organs, they're separate reactions, they do not share enzyme systems. They do not share membrane systems. They're separate systems entirely. They're in the same cell. Okay, I mean, plant cells will have mitochondria and chloroplasts right side by side, but these two systems do not share anything. That's why, once again, I don't like linking them too heavily together. Okay, let's take a break here because I need to drink some water and get my tongue untied from light independent and light dependent reactions. Okay, so 5 minutes or so, which is really 10, but 5 or 10 minutes, okay? A couple of things came up while we were on break, and I don't mean to draw attention to people in the room, but we found a couple of things. That is, there's a really good article that I shouldn't endorse it having not read it, but it looks like a good article. So if you just Google the color of plants on other worlds, it's really it's a Scientific American article, so it's got to be good, right? It's got good illustrations in it. I can't wait to cut and paste the illustrations, if nothing more. So you might want to check that out. The color of plants on other planets. Thank you very much. That looks good. It talks about the idea of the use of different pigment colors to absorb light and shuttle that on to photosynthesis. Deb had an app that you might want to check. It's free, you said, right? Called VCell on the Apple Store, on the App Store. Which has, it's about photosynthesis. It has a movie, it has a quiz, it has some writing material, it has some diagrams and so forth called VCell. You might want to check that out. And what was that site called?
This is on the NSF.
NSF? Not NASA. Oh, I thought it was NASA. Okay, on NSF, a really interesting article. How did you get to it? Did you Google something? Yeah, but you Google anything in particular?
I'm on the mailing list and they send.
Oh, okay, so you got it that way. Oh, well then can you forward that to me? Sure. It's Pratt at immaculata.edu, because I'd like to read that. Anyway, the article that I'm babbling about is about CO2 enrichment and its effect on plants. If it's the study I know about, they actually worked on large groups of plants. It's where they actually put out large tracts of pipe and pumped CO2 around the plants. It's big science. But they were pointing out that enhancing the CO2 levels of plants not only improved their growth, but how it did it was by improving the efficiency at which they used water. So their photosynthesis rate was going up, and their use of water was more efficient. They were using less water and using it more efficiently, which, as I was saying, and as an aside right before I started, that sort of makes sense because I know just from my botany experience that CO2, if you take a plant and give it some CO2, the stomates will start to close. That is, the guard cells will close, which would make sense then. If they close their stomates a little bit, they wouldn't lose as much water. They'd be able to retain water and use it more efficiently. They're getting the CO2 because it's an enhanced environment. The plants would be really happy. So it's really sort of a neat concept. So thank all of you for that. I like all those. I'll be getting the app. I've already got the article, and that's coming through my email. So that was worth it to me. Okay, I want to continue now with the light-independent reactions. That is the so-called dark reactions or Calvin cycle. I don't want to, just as was the case with the Krebs cycle, I'm not interested in each step. I'm interested in the concept that this is a cycle, that we begin and start with a substance. In this case, the substance we begin and start with, that is, which is constitutive in the chloroplast, is called ribulose 1,5-bisphosphate, or RUBP. That's the substance where the cycle begins and where the cycle ends. Here, the purpose of the Calvin cycle is to synthesize carbohydrate, not to break it down. Remember, in the Krebs cycle, the goal is to tear the glucose fragment, the phosphoglyceraldehyde, apart so that it can be effectively just dissipated as carbon dioxide, but energy can be harvested from it in the form of reduced NAD. Here, we're going to be synthesizing carbohydrate. It's done in a sort of unusual way, and we start with a 5-carbon and we add carbon dioxide, so we get a 6-carbon, but that's not going to last very long. It actually is fairly ephemeral, and then switches, does a couple of rearrangements, but almost, very quickly, that 6-carbon is broken into 2 3-carbons. The carbon that's ejected from the system, which is therefore the final product of the Calvin cycle, is phosphoglyceraldehyde, or glyceraldehyde 3-phosphate, G3P. So it is this 3-carbon molecule, not a 6-carbon sugar, as many textbooks say, but really a 3-carbon glyceraldehyde 3-phosphate, or phosphoglyceraldehyde. The balance, if you will, if that's the right side of the molecule, the left side of the molecule is then taken back and rearranged such that readjusted to regenerate the RUBP. So this cycle goes around and around, absorbing carbon dioxide, combining that carbon dioxide, and then injecting G3P through in the stroma, in the liquid of the chloroplasts, and that's the Calvin cycle. I'm not worried too much about all these. Now, this part right here will become important. That is this ribulose bisphosphate, the RUBP, and this addition of the addition of carbon dioxide to RUBP will become important, because this is, well, I won't write it up there, but is going to be catalyzed by the reaction referred to, or the enzyme referred to as Rubisco, which is ribulose biphosphate carboxylase. This enzyme is important because we'll see that it, there was a mistake made in the design of this. The Rubisco, the enzyme that catalyzes under normal circumstances the addition of carbon dioxide to RUBP, that enzyme, Rubisco, can equally catalyze the reaction of oxygen to RUBP. And that can be problematic, and I want to talk about that in a moment or two, but let me just finish this up. So this is a key step right here of this addition of CO2, and it turns out to be problematic in some plants. As I said, glucose is a 6-carbon sugar, and is, of course, mistakenly often listed as the final product in photosynthesis. It's not. It's G3P, that is glyceraldehyde 3-phosphate, is the final product. But you can quickly make a variety, you can't, but a plant can very quickly make a variety of substances from G3P, including glucose. So just by linking the two glyceraldehyde molecules together and monkeying with them a little bit molecularly, we can make the glucose, which is probably why it's said. I honestly don't understand why it became a convention to say that glucose is produced. I don't know where that came from, but it's been around forever, and it probably won't ever go away. But as I said, it's technically simply not true. The exit product is, as I said, glyceraldehyde 3-phosphate, or G3P. This process actually works. It is imagined that the Calvin cycle works in groups of 6, so that this cycle, so that the molecules are accumulated in groups of 6, so that you actually end up ejecting sufficient molecules and reestablishing RUBP and so forth. I think that's minor, but I'll mention it in passing. Once G3P is produced, it can be converted into all kinds of things. Glucose is one of them, starches and so forth. I want to address that in just a second. Photosynthesis is an astounding process to me. I mean, if you think about that, less than 1% of the Earth's sunlight makes it to the Earth's surface. That is less than 1% of the sun, or roughly 1% of the sunlight that starts in our atmosphere makes it to the surface. Of that, about 0.03 something, I don't know, don't quote me on that number. A small percentage of that is absorbed by plants, and plants aren't 100% efficient, and still we can make 155 billion tons of material per year. The plants do a pretty good job. Photosynthesis, although maybe not hyper efficient, but it certainly can get the job done. And I just think it's pretty impressive how that works. Okay. As I said, once you have the G3P, you can make other substances. You can make glucose. Then you can take the glucose and rearrange it and make fructose. And in fact, more plants are going to make fructose than glucose. That's the other thing I don't understand, why glucose was chosen. I honestly think it's probably just because everybody is so humanocentric. So of course plants are going to make the sugar that we need, because we're humans and we're special, so they're going to be making that for us. They don't, but that's the way we would think of it, and therefore keep propagating that concept. That's my humble opinion. But then you can shift that glucose to fructose, which is what most plants are going to make, not glucose, because they don't really want blood sugar. They want fructose, fruit sugar. Then you can take the glucose and the fructose and join them together and make table sugar, sucrose. Quite honestly, most plants will transport their sugars as sucrose. That's pretty good universal statement. That is, that sucrose is the way that the photosynthetic product is transported throughout the plant. That is, it's converted to sucrose and then moved through the phloem to wherever it needs to be taken to. Having said that, it turns out, though, that only really 2 plants produce sufficient amounts of sucrose to be commercially harvestable, and that is sugarcane and sugar beets. They produce such large abundances and store large abundances of sucrose that you can actually, get it out of them. And so that's where we get most of our sugar from beets, for the most part in the U.S., or sugarcane. That's not to say other plants don't. I mean, obviously, maple trees make sucrose, but it's nowhere near in the concentration of these plants, because think of how many gallons of tree sap they have to boil down to get a little jar of Vermont maple syrup. I mean, it's a huge amount. So my point is, sucrose is used as the preferred transport sugar, but only a couple of species really make it and keep it in sufficient quantities to be harvestable. Once you have that glucose or fructose and rearrange it to glucose, you can join it together to form starch, so that works real well. So you can have starch storage, as in this potato, so that happens a lot. There are other things that plants make, but the only other one that I'll mention is cellulose. Cellulose and starch, as far as building blocks, the monomer is glucose in both cases. The difference as far as chemically goes is really the arrangement of the glucose strands. In starch, they're largely linear with short breaks, side chains off, and they aren't generally interconnected very well. They're sort of independent molecules. in the case of cellulose, there's a much more parallel arrangement of the glucose chains into long chains, and then they're cross-linked by nitrogen bonds. So there's sort of nitrogen links in them, which is what makes them so strong. So if you look, you see you get the glucose strand, and then these will be put in parallel and then nitrogen linked, and that makes cellulose, which and lignin, which is very similar, pretty strong structures and resistant to really digestive activity, because we know that only a couple of organisms, a few bacteria and a few fungi, have the enzyme cellulase, can produce the enzyme cellulase and actually break down cellulose. It's very rare. It's a very rare enzyme. This is, again, I'm just trying to show that here are those strands, and then you can see some cross strands occurring, this sort of crisscrossing that occurs. So although they're very, like I said, starch and cellulose are made of the same thing, glucose, the molecular arrangement is quite different. We use that, of course, for paper, so this is just showing, again, the cellulose strands of high-quality paper. Okay, so I just mentioned that. Okay, I already recapped this. Well, let me recap this because then the next statement that I want to make. Again, the light-dependent reactions occur in the thylakoid membranes. The light-dependent reactions produce the reduced NADPH and the ATP by chemoosmosis. As a waste product, oxygen is produced by these light-dependent reactions, specifically in photosystem 2. Then the Calvin cycle uses the ATP and NADPH plus carbon dioxide from the atmosphere to run a cycle where it synthesizes G3P, glyceraldehyde 3-phosphate, to not glucose, and then that G3P is then organized the way the plant wants. It can even take that 3-carbon chain, rearrange it, aminate it, make an amino acid out of it if it wants to. I mean, plants are very metabolically agile, so they can do a variety of things with that simple 3-carbon skeleton. Okay. That other stuff is just sort of verbiage because I put it there. I just wanted to mention in passing that oxygen byproduct is pretty important. We certainly expect that to be the case. Remember that prior to the rise of photosynthetic plants, which, well, if you include the cyanobacteria, we're somewhere around 3.7, 3.2 billion years ago, somewhere in that neighborhood. That's if you still go with 4.5 billion as the age of the planet. But in that neighborhood, prior to that, no photosynthesis, virtually no oxygen in the atmosphere. Then with the birth and the radiation of of a variety of photosynthetic plants, oxygen became a fairly high constituent of Earth's atmosphere, around 21%, and it's constantly being replaced. Most of our oxygen is replaced by algae, not by terrestrial plants, but they still play a role in it. And so it's a waste product of photosynthesis, but obviously pretty important to the animal kingdom. Okay. Now, what I've described thus far, these plants that add CO2 to Rubisco, or add CO2 to RUBP by using the enzyme Rubisco, are called C3 plants. They produce a 3-carbon. The prevalent product is a 3-carbon molecule. It's actually the G3P, and so they're called C3 plants. These are the bulk of the plants bulk of the plants on the planet. 85% of all plant species are C3 plants. They include wheat, rice, barley, all those that I've listed there, tobacco, spinach. Most all the trees that we come in contact with are going to be C3 plants. Most lawn grasses, except my yard, is all crabgrass, and so that's not a C3 plant, but everybody else's lawn, and I think dandelions are C4 as well. So my entire lawn consists of crabgrass and dandelions. That's good because, you know, they sort of take over at different times. Crabgrass later in the fall, or towards the fall, the dandelions in the spring. It's nice. Anyway, most of the world's plants fall into that category. The problem, as I alluded to before, with C3 plants, although I really don't like to call it a problem, because, I mean, I call it a problem because I guess it's a problem, but they seem to do okay. If 85% of all plant species are C3 plants, you can't really call it a problem, right? If they're all surviving and thriving and so forth. But anyway, if there's a weakness of C3 plants, the weakness is the process of photorespiration. Photorespiration is the term that's used to describe the process. It's a competitive reaction, but the process where Rubisco, instead of adding CO2 to RUBP to start the Calvin cycle, instead of doing that, which is what you want to have happen if it's photosynthesis, what happens instead is the RUBP is oxidized. That is, Rubisco adds oxygen to RUBP, breaks it down, releases it as forms of carbon dioxide. No energy is produced, no sugar is produced, that is, no carbohydrate is produced. It's basically, you could think of it as a cannibalistic reaction. That is, the plant is destroying the RUBP, the ribulose bisphosphate, that it needs for the Calvin cycle, and it's getting nothing for it. Okay? The sad part is that it's a simple competitive reaction, and so as CO2 levels drop or O2 levels increase, then the competing reaction, the addition of oxygen to RUBP, will also increase. Now, how much of an impact that is that? Estimates indicate that it's probably close to 50% of the potential photosynthate that a C3 plant makes is lost in photorespiration. A huge amount. Even if it was only 30%. If it was 25%. That's a huge amount of loss of energy harvesting that's happening because of this competitive reaction. Now, I don't want to make, as soon as I say that, make a huge deal of it, because these plants, you know, those trees out there, they're doing just fine, thank you. Well, okay? But one can see that if that problem could be addressed, that is, the elimination of photorespiration, then you could greatly enhance the we'll call it efficiency of photosynthesis. You could greatly enhance the photosynthetic product that was produced under the same conditions. Evolutionarily speaking, plants have found two solutions to that photorespiration problem. They are referred to as C4 plants and CAM, which stands for, don't ask me to spell it, crassulacean acid metabolism, CAM. CAM plants, okay? Now, C4 plants are really the ones that I want to talk about here. Crass plants, that is CAM plants, are I'll mention them, but their overriding issue, the CAM plants, is drought stress. Because if you notice, they're desert plants. They're cactus and wax plants and some others that are adapted to pretty harsh conditions in many respects. Not entirely, but in most respects. So CAM plants and a lot of succulents, and they're really addressing the water issue as much as the photorespiration issue, but it's a sort of bonus. They get a bonus out of this. Now, The C4 plants, also sometimes called the Hatch-Slack pathway, okay, Hatch and Slack, they sound like a comedy team, but they were really biochemists. They discovered this pathway. And as you can see, it's a relatively small number of the known species of plants, about 0.4% of plants, but they're fairly common named plants, if you'll notice. Corn and sorghum and sugar cane, crabgrass, Bermuda grass, a lot of sedges are C4. As I said, I believe dandelion is. I know that purple loosestrife is. I know that water hyacinth is. I know that I can't think of the other plant. Kudzu, all those are as well C4 plants. C4 plants, well, they found an interesting thing to do. Okay, that's C3. I don't want that. C4 plants have solved the issue of photorespiration by physically separating this process of CO2 addition to RUBP. They have physically separated the process. What do I mean by that? Well, the first thing that C4 plants have done is rearrange their leaf tissue. In C3 plants, the inner layer of the leaf, called the so-called mesophyll, and then the layer right above it, the so-called palisade layers, that is the middle two layers of the cells in cross-section of a leaf of a plant, are organized in nice columns in the palisade layer, and then sort of a spongy billowing layer called the mesophyll below it. But in any event, they are arranged in such a way that the carbon dioxide that's used in photosynthesis is fixed, that is added to RUBP right in the chloroplast stroma, right where the Calvin cycle is located. The C3 plants have rearranged, that's C3 plants, the C4 plants have rearranged plant tissues such that the first step is to take the carbon dioxide and fix it and make it into another compound. That compound is probably on here as oxaloacetic acid, yeah, OAA, oxaloacetic acid or oxaloacetic acetate. Oxaloacetic acid is a 4-carbon molecule. So what the plant does is simply attach carbon dioxide to an organic acid and makes oxaloacetic acid. It does this along in the area next to the vascular tissue called the vascular bundles, not that that matters, but in a different place. So this fixing of carbon dioxide to make oxaloacetic acid occurs away from the palisade layer of the cell where the Calvin cycle would be located. So they're physically separated. What that allows the plant to do is make oxaloacetic acid, add, in essence, convert the carbon dioxide to oxaloacetic acid, then transport the oxaloacetic acid to the cells, and that's what this is trying to show here, to the chloroplast, to the cells where photosynthesis are taking place, and then donate that carbon from oxaloacetic acid, not from carbon dioxide directly. That was done up in the bundle sheath cells. But now we've separated that process. We have a preferential sort of direct line for connecting the carbon to RUBP. Photorespiration is virtually eliminated. It might not be 100% effective, but it's pretty darn effective. These plants automatically now gain all that photosynthetic product that would be otherwise lost due to photorespiration. In other words, they're independent of oxygen concentration. C3 plants run into this problem of photorespiration every day because as they're photosynthesizing, they're producing oxygen in their very tissues. That oxygen accumulates and starts to poison the photosynthetic reaction. That's another reason, or many reasons, I mentioned moisture stress regions, why I say those trees out there are done photosynthesizing for the day. Even though it's a beautiful day, they're not photosynthesizing anymore. They switch to respiration because they can't handle all this of what's going on, because that's a C3 plant. Oxygen levels built up. They're losing, so they just shut down. They're losing water. They just shut down. They're not photosynthesizing anymore. They quit 15 minutes ago. Now, I don't know exactly how long ago, but they do. Plants do that all the time because they have other things. There's a compromise going on. So C4, the key is that C4 plants have separated physically within the leaf the fixation of carbon onto oxaloacetic acid, and then that donation of the carbon to RUBP, still done by Rubisco, but the addition of RUBP, and that pathway, because it involves a couple of steps, because it uses either a malic or aspartic acid as the root, and then adds the fourth carbon on to make oxaloacetic acid, discovered by, like I said, Hatch and Slack, and so it's sometimes called the Hatch-Slack pathway. Or C4 plants. Okay, it's named C4 because oxaloacetic acid has 4 carbons. So that's how C4 plants work. And okay, this is showing the same thing. That is, so we physically separate that process of carbon fixation from the Calvin cycle. Now, CAM plants, that is the crassulacean acid metabolism plants, have other things to consider rather than just photorespiration. They deal with desert-like conditions. So if we focus on cactus, although other plants, other succulents could be used too, but if you think of a cactus out in the sun, in desert, can get very, very hot, can be very, very dry. They've done a lot of things externally to take care of that moisture problem. They've gotten rid of their leaves, used just their stem for photosynthesis. They've made them, well, they haven't gotten rid of the leaves, they just made them into spines to protect the stems where the photosynthesis where the photosynthesis takes place and the water is stored. So they've done some external things. Internally, what they've done is separate carbon fixation temporally, not spatially. C4 plants went in the direction of, evolved in the direction of physically separating, spatially separating carbon fixation from the Calvin cycle. What CAM plants do is temporally do that over time. What you find happening is there's a day-night cycle in CAM plants that seems counterintuitive at first. At night, these plants open their stomates and close their stomates during the day. A C3 plant or a C4 plant would open the stomates during the day to allow carbon dioxide in and oxygen out, and close them at night to just protect themselves. These plants do just the opposite. They open the stomates at night, or stomata if you prefer, stomata at night, to absorb carbon dioxide, and then close them during the day so that they don't lose water. So that's the cycle. What they do is similar to what Hatch-Slack pathway plants do. That is, they collect the carbon dioxide, in this case, in a variety of different, depending on species of the plant, of different metabolic acids. Malic acid, fumaric acid, oxaloacetic acid, probably some others. They're the only 3 I can think of right now. So what happens here is at night the carbon dioxide is absorbed into the plant, and here they're using a little different substance. They're using phosphoenolpyruvate, but it's the same sort of overall idea. The molecules change, but and if this was a botany class, or plant physiology class, we'd worry about that. But the carbon dioxide is then combined, so we get a 4-carbon molecule. Here they're calling it malic acid, but it could be fumaric, succinic. Would be another one that could be used and is used depending on the plant. Anyway, now we accumulate generate these metabolic acids overnight, which then can be broken down to donate the carbon into the carbon cycle during the day when photosynthesis takes place. The plant doesn't have to have the stomata open because, and therefore reduces its water loss, because it's sort of gathered up the CO2 at night, which is pretty good because it's not only gathering the CO2 that it's producing in respiration, but other organisms around it at night might be respiring as well, and they can pick up the CO2 from that. So CAM plants separate this carbon fixation by time, temporally, as opposed to Hatch-Slack pathway or C4 plants, which separate this process temporally. So there's temporal versus spatial. This is actually C4 plants again, but I don't need that anymore. So in summary, then we have these three kinds of plants in the world: C3 plants, C4 plants, and CAM plants. And they all do the same thing, that is fix carbon dioxide to make phosphoglycerol or G3P, phosphoglyceraldehyde 3-phosphate, in the Calvin cycle, but the difference is how they deliver the CO2 to the Calvin cycle. See, the traditional C3 way is to just direct add the CO2. The C4 plants add an additional step of using oxaloacetic acid to donate the it doesn't actually have to always be OAA, but it almost always is. I don't know of plants that don't, but I'm sure there's one out there that uses something else because it's just a 4-carbon acid, organic acid. And the CAM plants separate this process over time. And so they've met the challenge in a couple of different ways, which I find sort of interesting as well. Okay? Okay. Any questions or comments? Okay. Yes? No? Okay. Okay. What I'm going to do is push it a little bit more, take another little break, and then we'll be able to bring it home. Okay. What I can't click and talk at the same time, so give me a second here. Come on. Okay, I always like, you know me, I like to use a case study. And at first this case study might seem as if it's not really related, but it is. This is a case study that looks at the problem the plants have to deal with environmentally, that is, certain stresses, and how, if I were an agricultural scientist, I might want to help these plants. Now, because if I could find a way to deal, for instance, with the different kinds of stress, cold stress, that's frost damage, or heat damage, or salt damage, or even salt damage from evaporation and irrigation, if I could find a way to help the plants withstand these stresses and these plants would continue to grow, you know, I might be able to make a few dollars. And so the idea is, we know plants vary in their ability to withstand these stresses. Is there anything that we can understand about how they do that, and then we can genetically modify or otherwise manipulate plants to improve this stress? Well, there is. It turns out, and this is a little bit controversial, in that what I should say is not universally accepted by all plant scientists. I guess is what I mean. It's not controversial in the sense of, you know, politically controversial. There is this chemical, glycine, betanine, or I'll just keep calling it glycine because I seem to be having trouble speaking. This chemical is produced in plants naturally under stress. Okay? There's been a lot of work shown that it's produced where the stress occurs, it is transported throughout the plant. which is where the controversy comes in, because some scientists think this is not a chemical that will prevent stress, but is a chemical that is produced in response to a stress. But I'll show you some examples where it certainly gets muddied as to what's going on. Okay? Now, there are a couple ways that this could be worked out. One way would be to take this and spray it on a plant. And there's been a fair amount of research done with that too, and I have to say the results on that are disappointing. Okay, they don't give very clear-cut examples. I mean, what you're expecting to happen, or what you were hoping to happen, or what these folks who did the experiment were hoping to happen, is that they could take this glycine, because it's being produced by stressed plants, and they could spray it on plants and then freeze the heck out of them, and they would do okay, or subject them to drought stress and they would do okay, or heat stress and they would do okay. It was, some plants seem to respond, some don't. It's probably not so much that it's whether it's the glycine or not, it's probably more of, is it getting through the epidermis of the leaf or not? Because, some leaves have a lot of wax on them and some don't. So they tried surfactants, to dissolve that away. Well, that's problematic, because then that increases drought stress, because you're removing the wax and so forth. So the topical application of glycine made a big splash. Monsanto was all excited, and then they stopped. Maybe that's why they sent those genetically modified seeds to Oregon or wherever it was that they did. Okay, by accident, they say. Anyway, the other way to do this is perhaps to genetically modify the plants. That is, we know what glycine is. It's an amino acid. This is just a little bit of an adjustment to glycine, but we know that we know what the sequence, we know what the substance is. We know what the sequence is. We can insert that sequence in it. We can put it in a virus. We can transfect the cell. Life is good. We can make genetically modified plants that are transgenic plants that have the ability to produce the glycine. I'm not going to keep saying the benadine because I can't say that. And here's an article, okay, genetic engineering of the glycine synthesis in tomato, how it protects flowers, chilling damage, drought, or in this case, this is chilling damage. Whoops. What I want you to see here is what was done was these experiments where we have pictures here. And they might be a little hard to see, so I might have to interpret them for you. These are wild-type plants. These are transgenic plants. And what was done was these plants were, these are just different plants to sort of show the example of what took place. They were exposed, as I said, to temperature of 3 degrees C for 7 days, and then transferred to a warm greenhouse to allow to continue to grow, and then they looked at them to see how they grew. And this is transgenic, and these are wild type. Wild type means just a natural plant, right? These have the glycine gene, these don't. You can see a difference. Okay? These are wilted and drooping. These are healthy and good looking. These have flowers. These don't. Looks good. Okay? So starting to look good. I already answered that question. Wild type is the unaltered group, so I don't care about that. Okay, I don't know why I put that there. Well, I know why, because I use this in class. So, okay. What I would basically ask, I think I basically answered my own question here. This is the same slide again. After exposure, you see what happened. The glycine helped improve the maintenance of the leaves. It kept the plant healthy enough for it to actually flower. You can see a lot of damage here. The leaves are withered. They're nice and healthy on both of these plants. These look like they're frozen to death. So I already answered my own question there. We see that. So here's a graph of these data to look at a little differently because I want to say a few things about them. What we have along here is the days of after cold treatment. So again, remember, they were placed in the cold, so they were given the shock of cold in this case, and then they were moved to a greenhouse for observation. So they were observed in 1 day, 3 days, 5 days, 7 days. This is a measurement that we took, and I'll get to that. It's called ion leakage. That is, what we're looking at here is how ions are leaking through the membranes of the cells. It's a fairly difficult measurement, but these folks were able to do that. And what we have here is wild type is blue. And these are two different transgenic lines, but they're both transgenic lines, so they both have the glycine gene in them. And I think you can agree with me, there's a difference there. They even have their nice error bars. What you see is that these guys are experiencing ion leakage, which is what's causing those leaves to look all droopy and all, because the cells are oozing inside, because the membranes are broken, and so the ions are leaking out, and so we see that leakage continues for several days afterwards. I mean, it levels off because I guess there's only so many ions and so many membranes that can leak. I don't know. Maybe they're trying to heal themselves. I don't know. And here, the ones that have the introduced glycine gene, they have less leakage. Okay? We saw pictorially that they were better. Now we see on a cellular level that these cells or these plants are better. Okay, so that's looking good, I think. I'll ask this. You don't have to click if you don't want to, and then I'll sort of answer it myself. But I'll give you a moment to think about it. Which photosynthetic process? By that I mean Calvin cycle, ATP synthase, movement through the cytochrome system. That's D. Light capturing photosystem. Which do you think would be sort of a gut reaction? You know enough to answer this question if you think about both respiration and how that works, and photosynthesis and how that works. I mean, this is photosynthesis, but just about how cells work. Which one of these systems in photosynthesis is most likely to be impacted, do you think, by this ion leakage, the breaking of membranes and allowing ions to freely pass through them? Any idea? Well, this one is proteins and pigments embedded in the membrane. Doesn't really rely on any ions, it relies on photon. So I don't like that one. The Calvin cycle, it's free in the stroma of the chloroplast. There's no membrane involved, so I don't like that one. ATP synthase is dependent upon there being a membrane to keep the protons in high concentration so that they can run down a hill. I like that one. Okay, and I don't even know what that one means. Whether electrons will transfer from PS1, I guess, to PS2, or from PS2 to PS1. That could be vaguely affected, I guess, because they're in the membrane, but they're not ions again that are moving around. They are electrons. So the correct answer is C. That is, it's ATP synthase is the one that's being affected here. And that makes sense. Now we understand in that little exercise how frost and heat, as well, as we'll see, it's the same thing, but how frost damage affects a plant. It screws up the thylakoids. Okay, they can't establish the proton disequilibrium. ATP synthase can't operate. The plant starves to death because there's no ATP to make, or it can't make any ATP. Okay, well, I thought that was interesting. Okay, so what else can happen? What about heat damage? Okay, that we did cold damage. What about heat damage? Okay, here's another article. Genetic engineering and biosynthesis, again, of the same thing. I know that I'm calling it just glycine. Enhances thermal tolerance of photosystem 2 in tobacco plants. Okay, so basically here, let me expose everything here. I don't know, I have that on reveal. This is looking at changes in oxygen producing activity of photosystem 2. Remember, photosystem 2 is the one that operates photolysis. So it breaks down the water to form oxygen. So we're looking at the oxygen producing activity of PS2 determined by thylakoid membranes isolated from leaves after exposure to different temperatures, the temperatures or in chambers for 4 hours in wild type and transgenic plants. The values are means, blah, 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 blah. WT is wild type, so the red is wild type. The other color, whatever that is the genetically modified transgenic plant. This is temperature on this axis, and this is oxygen activity isolated to photosystem 2, that is photolysis. And what do you see here? We see that the wild type loses its ability, to photolyze, to break down water to form oxygen, right? Because this is oxygen evolving activity of PS2, and as time goes on, the ability of those wild types, which is the red one, declines dramatically. Whereas those transgenic plants maintain that for a while. Yeah, eventually they're going to run into problems, okay? But they sort of hold their own for a little while. This may not be as dramatic as the cold stress experiment, but it's kind of interesting. Okay, so what did I ask there? Like I said, I use this for students, so I'm trying to see. Okay, produces O2. Well, PS2 does that. Okay, I already said that, so I don't know. That was stupid. Okay, so we'll just move on here. Let's look at this. This is the same data again. Then this is again this oxygen, and I basically asked you to try to make a couple of observations from this. I'll save us some time by just the things that I sort of saw from that. That is that colder temperatures. Let's see, yeah, the colder temperatures there was no effect. That is, you couldn't see a difference between the two at colder temperatures. It's only at the warmer temperatures that you started to see a difference in the oxygen generating capacity. So so the difference didn't become noticeable till then. That the wild type was less than the transgenic type. I don't know if there's any other observations you could make or not. One line's red, one line's blue, that sort of thing. But again, I just wanted you to see, again, not necessarily you, but my students have difficult time interpreting graphs. Anytime they see a graph, they shut down. So I spend way too much time having them look at pictures because I think it's important. Okay. This is just showing, because it sets up the next question, this is showing a more complicated, it's hard in just a sketch to show a photosystem. This is trying to show photosystem 2. The idea here is that we're going to take this a little bit and look at it from the standpoint, I don't know why that thing won't stay out of the way, of which part might go through the membrane and which part is sticking out from the membrane, and I don't know. I'll just say the blue box is what sticks into the membrane. That's the transgenic part. This is the part that's sort of sticking down into the surrounding environment of the thylakoid. That becomes important because I want you to see how that would. How that would affect things. So the blue box is transmembrane, and these red boxes are adjacent to the membrane. They adhere to this, so they're on the membrane but not going through the membrane. That'll become important later, so I'll mention that. Oh well, let's strategize about it right now. Okay, knowing that little bit, that 2 seconds about molecular membrane structure, which of the following could explain the increased temperature decrease? Which of the following could explain why increased temperatures decrease PS2 activity? Remember, we found that increased temperatures cause the oxygen evolution from PS2 to go down in really both transgenic and wild type, but certainly in wild type. Would it be the thylakoid membranes become more permeable to ions, the chlorophyll breaks down, the peripheral proteins lose their ability to bind to transmembrane proteins, or water cannot bind to PS2 to form oxygen? Well, again, I'll save you the pain. It's C. It's the idea that what's going to happen is those peripheral proteins can't stay attached. The transmembrane proteins, I guess you need to know more about transmembrane proteins. Transmembrane proteins, by definition, are highly embedded because they pass all the way through. They're usually double loop, 7 loop proteins. They got a whole bunch of names. 11 loop proteins, that's the number of loops that they go through to sort of like lasso themselves and lash themselves into a cell membrane. If you're dislodging transmembrane proteins, you're using nuclear weapons to do that. I mean, you're really disrupting the entire membrane, the very foundation of the membrane. Not really gonna happen and still have the plant survive, or any organism survive to any great extent. I can think of some, I can debate with myself about that a little bit, but that's certainly the case. Proteins that are just adhering to the inside or outside of the membrane can be much more easily dislodged. And that's just logical to me that would be the case, because they're not embedded in there. They're not wedged inside the membrane, okay? So the most likely explanation for this is this idea that these peripheral proteins become detached, okay? When you heat the system. So we see in frost damage, glycine stabilizes the membrane or prevents the membrane leakage. In heat stress, glycine appears to help maintain the peripheral proteins on the thylakoid membrane. So we have 2 modes of activity of the glycine in helping plants. Now let's look at what happens in salt concentration. Here's another article. This one's, I think, the oldest of the 3. But here, this is looking at the idea of what's happening with salt concentrations. So, well, first, actually, it will ultimately look at salt concentrations. Right now, we're looking at the concentration of the benidine, that is, of the glycine along the x-axis, and the amount of protein that remains associated with the membrane on the y-axis. This is kind of hard. I need the yellow filled in here. Okay, I don't care if we make 2 observations. You got to have a way of measuring the size of proteins. An Dalton is the sort of size measurement that's used for proteins, and these are kilodaltons, so they're just, everything's multiplied by 1,000. So we have units. We have a 18 kilogram unit, a 23 kilogram unit, or kilodalton unit, and a 33 kilodalton unit. And we see that we get a different pattern here, right? Again, if I give you, like 2 seconds to look at that, what you're going to see, I think anyway, is that the size of the protein has some influence, okay, on the ability of that protein to remain attached or associated with the membrane, right? As the size increase, larger amounts of the protein remain adhered. The way I would look at that is, in a simplest form, I could get into a little trouble with it, but is that the bigger the protein, the more space it has, the more ability it has to attach at multiple points to the membrane. So the bigger the unit, the more contact points it can have with the membrane, and so it remains attached. Now, if you notice the amount of, so we see that happening. We also see that the more of the glycine that we add, the going in this direction, the more adherence of each of the three that can happen. So this helps keep those these peripheral proteins attached to the membrane. Okay? So we're building a series of experiments here, or we're culling a series of experiments here that give us a picture of why this works. We see that it works because we, as I said, and we know that plants naturally produce this substance, and now we're demonstrating in various modes that this can actually cause less ion leakage, better attachment of peripheral proteins, less detachment of peripheral proteins, and so forth. Okay. So the idea here is that this glycine, because as far as this research goes, basically the idea is that it protects the membrane within the thylakoid membrane against excess salt. Remember, salt's going to build up either because of salt from road salt or something like that, or salt that could build up because of irrigation. But the idea is that what it does is the glycine protects the plant because it protects the membrane of the thylakoids. I know I can say this, such that you don't have dissociation of those peripheral proteins, so the process of photosynthesis can continue. Okay, that could have been better said. Anyway, therefore, this whole system has to be together, and glycine manages to protect a variety of different parts of the photosystem to maintain its integrity so that it can operate. That's what I wanted to get from that by looking at those actual research papers and pulling pieces out from it. Okay, so that's that. The next thing, I have a series of short clips, or not clips, but quips here I want to go through. This is another exercise to get people to think about what's going on in a plant. The idea here is we're going to take this plant that we can put in a chamber here. It's a little hard to see, but it's in a glass chamber here. That's the pot. There's this bean plant in here. It's a closed chamber, and this is a CO2 sensor. So we can measure carbon dioxide at parts per million inside this chamber. The idea here is to record changes in carbon dioxide over a long period of time when the plant is exposed to different light conditions, and then see, because we know that CO2 is used in the photosynthesis, right? It's used in the light independent reactions of photosynthesis, the Calvin cycle. So in essence, if we measure CO2 uptake, we're really measuring photosynthetic rate. here's some data that were collected using that apparatus that was just described. Down here is just a time frame. Okay, it happens to be in seconds, thousands of seconds, but it's in seconds. And along this axis is the carbon dioxide level in parts per million. And these just mark events on the chart. A is when the plant was introduced into the sealed container. B is when the light was turned on. So it's now being exposed to light. C is when the light was turned off and it was actually wrapped in black paper to keep light out, so that's C. D is when the black paper was removed, and E is the end of the experiment. And my question is, what's going on in with respect to this plant photosynthesis, or CO2 release, or uptake, I'm sorry, I apologize, uptake and release, but the CO2 levels and this plant. What's going on? Put the plant in, so what's happening between A and B? A is when the plant was put in, B is when the light was turned on. So what is that showing us that's happening? It's CO2, right? CO2 levels are going up. It's respiring until the light's turned on, right? So this represents respiration. It's not a line, so I'm sorry, but this would be respiration, right? Because CO2 levels are going up, the light hasn't been turned on, the plant's in there, it's not photosynthesizing, or it might be doing a combination of it, but the net is more respiration than photosynthesis because the CO2 levels are going up. Right? Sure. Okay. Now, here, B, the light is turned on, so CO2 levels drop. It's photosynthesizing. Good, right. C, the light is turned off and it's wrapped in black paper. So this would be respiration again. Okay. Now at D, the black paper is removed, so there's a little plurp here, but I mean it's still in the dark. So all that, I'm not sure what that is in this experiment. I really don't think it was photosynthesis because it's still in the dark. Maybe it was just the instrument when they banged it because they were taking the paper off. You know, and if you jostle some equipment. Well, that's a possibility I didn't think of. Just turn on the light to take the paper off and then turn it off again, and so maybe there was a little thing. In any event, they don't tell us what it is, but that would be a good answer. Yeah, I mean that would explain why that is. Okay, so that's just a simple little experiment that allows me to see whether you understand what's going on. And remember, well, the next one will show this even better. But remember, these processes. Both take place, these processes, photosynthesis and respiration, both take place in a plant, okay? They can even take place simultaneously. I can't click and talk at the same time. Here I am. Is this what I want? Whoop. Like I said, I can't talk and do this at the same time. Jesus Christ. Okay. Because I'm tired. And I'm about ready to start cursing, you know. Not that you haven't heard it, but you don't want to hear it. Okay, let's see. What the hell is wrong here? Now, okay, it's gonna work this time. Okay, I minimized something, and I didn't want to do that. Now, oh, I know what I'm doing. See, it is late. What the hell? That's not what I want. I want this. Jeez. Okay, I'm back now. Okay. Now, I think this is the one I wanted. Yeah, okay. This is sort of pictorially an experiment, okay? So, this first is the demo part of it, and then the other part is to interpret it, which see if I can get that PowerPoint to come up. Okay, one way that we could look at photosynthesis is again that CO2 concept. We know that, and respiration as well, we know that if you add CO2 to water, it will lower the pH of that water because the CO2 will interact with the water, formed bicarbonate ion, and then carbonic acid, and it will acidify the water. And so you see where I'm going with that. We can use pH indicators to sort of get a, if not a quantitative, at least a qualitative idea of what the level of CO2 is in a particular sample. Okay, so here, this is just to start, is the pH indicator in little vials and little bottles that indicates, okay, that this is sort of moderate levels of CO2, so the orange and the reddish in that area. As you add more CO2, it's going to become acidic and it'll show up as yellow in this pH indicator. And if you extract CO2, take CO2 out of that solution, the solution will be more blue, violet, purple, whatever color that is. Okay, so we have that indicator that we could use. So you don't need to read all this, but basically we're going to take this container, take a container, put the indicator in it, put a plant that's a lot like Elodea, an aquatic plant in it, seal it, and take one of these and put it in the dark, one of these and put it in bright light, one of these and put it in a little bit of shade, and another in deeper shade, or something like that, okay? And then we're going to look at the results, okay? So keep in mind that green plants and algae use up carbon dioxide, removing it from the indicator as they carry out photosynthesis, but they can also produce carbon dioxide as they respire, okay? And there's my color chart again, okay? So the idea is, what color would the indicator become if you breathe out into it? Of course, it would become more yellow because you're adding CO2 to it. What color would the indicator go? That's bad English. It fits for right now. Indicator go if the plant was respiring and carrying out photosynthesis at the same rate. Well, that's a good question. If CO2 was balanced, where do you think it would be? CO2 was, I didn't say that right, where if photosynthesis was balanced and balanced with respiration, if photosynthesis and respiration was balanced. I need a drink, I don't need water. Okay, that would straighten it out, I know. The point here is that what would happen there, that is, it would be both making CO2 and removing CO2. So there would be a moderate, sort of like what happens in this atmosphere, right? Plants are taking CO2 out, we're putting CO2 in, so we would fall in this range, okay? See, that was easy to say. Okay, now, that last one doesn't make any sense. Okay, so here are some results, and then I want you to do some prediction in a second, okay? Here is a vial that was put in bright light. Sure enough, decreasing CO2, it turned purple. It's removing CO2, makes sense. Here's one that was put in some shade. So some CO2 is being produced, some CO2 is being consumed, some photosynthesis is occurring, some respiration is occurring. So it falls in this sort of reddish area here. Whether it was this one or this one or this one, I don't know, but it would be in that range, okay? Here's if you put it in the dark, okay? You get a sort of a greenish yellow, so it's down in this, well, green, it looks more green than yellow, but that's because there's a green plant in there. So you have to sort of look down here, and it's more sort of through it, and you can see it's sort of yellow, so it's down this way. It's respiring, so it's putting more CO2 into the indicator, okay? So we have that part of the exercise sort of taken care of. Now, I called it quiz, but it's not really a quiz. But this is a chart. Now, I stole this chart. I stole some of this from a British site. So this part they use the term hydrogen carbonate indicator. It's pH indicator. I mean, they're so quaint. Okay, so they called it that. And so the idea is that you're supposed to take these colors and match them to the particular unit. That is, that you take, you have the indicator and pondweed placed in light, 20 centimeters from a lamp, what would happen? So it'd be probably somewhere over here, reddish, bluish, purplish, right, over here, because it's definitely going to be photosynthesizing. It's going to be taking carbon dioxide out. I'll pop the screen up there in a second. Okay, and then you have the indicator, the pondweed, surrounded by a thin layer of shading. Over here it's explaining that that's when respiration and photosynthesis are both taking place. That's more of over here by the red, right? Taking some out, putting some in, so it's going to be sort of balanced. Then we have the indicator pondweed surrounded by a thicker layer of shading. So we're going to get darker. It's going to push us more into respiration, so it's going to be over in this level, right? And then finally, you have the idea of the indicator and the pondweed placed in a cupboard, that is under a cabinet in the dark, going to be way over in this direction. So when you're done, you can sort of put the jars in. That is, you get a sort of purple up here because photosynthesis is taking place and decreasing the carbon dioxide. You get a sort of reddish, and again, I don't care which one of these you would pick, but it would be in this sort of level because some CO2 has been added, but some's being taken out, so somewhere in that range. Here, we're getting closer to the idea that there's a thicker layer, so we're moving. Actually, I think I should have picked a little more orangish now that I look at it. If you want this, I'm going to put it up on the wiki, so you'll be able to actually get it for what it's worth, okay? But you're welcome to take pictures of it now. I didn't mean to interrupt that. But you might get me in it, and I don't want yeah. Well, you know, because this is my Alfred Hitchcock impression. Boom, 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 boom. You know, I sort of look like him from profile in a shadow. Anyway, the point here is that we see that it's going to be in that sort of orangish, reddish over here. If you wanted to pick that one instead, you can. I mean, literally, I just cut and pasted them electronically in there. I mean, so in second thought, I would have picked that one, I think. And now then this one, where you have lots of respiration taking place, you're going to push it way over in that direction. If you don't like doing it that way, you can also do it this way. That is, you could simply give the contents and the color, or you could even cut and paste the color in here, and then ask to pick the explanation, if you like that better, so that you would end up coming up with this, that is, the respirations, etc., is taking place, not taking place.
So if you have a pond that has a lot of like water lilies and algae and pondweed and all that kind of jazz, there's a lot of photosynthesis taking place during the day.
Yep.
So it's sucking out the carbon dioxide out of the water, and it's going to be rich in oxygen. So that's good for fish.
Yep.
But at night,
yeah, I see where you're going with that. At night, they are going to remove, those plants are going to respire, and they are going to remove CO2, or they're going to remove oxygen and produce CO2. pH will fall. It might fall depending on the size of the pond and temperature conditions and so forth. It could drop, you know, the pH could drop 1 or 2 units. I mean, it could be a significant.
would that negatively be affecting the fish population?
Well, generally speaking, the plants will, you know, unless you had a long stretch of very dark days and very long nights, which occur perhaps in the fall or in the spring, the plants are going to produce so much more oxygen during the day that it'll be a net gain for the pond. There'll be more than enough to make it through the night with all the respiration going on. Okay, because the plants, again, I hesitate to say it because they're not competing reactions, but photosynthesis runs faster than cell respiration. I mean, if a cell biologist heard me say that, they would get mad at me. or a physiologist, but the idea is that the products produced from photosynthesis generally outweigh the respiratory costs of a single plant, then multiplied by multiple plants. There's a net gain of oxygen and CO2 in a particular local area. Now that balances out globally. That's why it's 21% and not 21 today, 22 tomorrow, 23% oxygen the next day. But it is why it increased from 0 to 21% over a billion years, okay, or a couple billion years. So again, just to recap on that, the idea is that the net would be that there would be more than sufficient oxygen to make it through the night, even with the respiratory costs. Where you might have a problem would be, as I said, if it was a huge fish population, if there was really dark, cloudy, dreary days, several in a row, and really long nights. Then you might tip the balance enough to actually cause a real depression of oxygen and a depression of CO, a change in pH as well. Okay. Any other questions? Okay, just to let you know, tomorrow what I'm going to have you do, so that you know, is I will have to, just a little introduction of about 5 or 6 minutes, and then I'm going to task you with designing a seed, okay, is what you're going to do. I know it's totally different from this, but it's an actual activity. We're going to talk about seed dispersal, and you're going to construct the seed, and then we're going to throw them out the windows and do some other things and see if you can design a decent seed for dispersal. So that's what we're going to do, okay? So.